Welcome to Life and Biology. I'm Dr. Joel Graff. In a previous video, which I'll link to before, we talked about how cells begin to defend themselves against a virus infection. And the main take home of that video was the idea that once a cell realizes it's infected, it produces all sorts of new genes to provide that protection, such as antiviral genes. Well, changes in gene expression aren't the only thing that can happen during the, transcription of genes isn't the only thing that can happen in response to a virus infection. Another thing that can happen is that the cells can try to do things like shut down translation across the cell, because remember, viruses are essentially parasites of the host's translation machinery. So when a cell gets infected with a virus, there's a couple things that the cell could possibly see, and we'll talk about what happens downstream of that. One is that usually in a cell you don't have long stretches of double-stranded RNA, but when we learned about all the different virus genomes, we learned that there was some virus genomes are just made of double-stranded RNA, and others there's a double-stranded RNA intermediate while they're trying to get copies of their genome. And so double-stranded RNA would be pretty good indication that you have a virus infection. So there's a protein called protein kinase R. The K kinase means that it's an enzyme that'll phosphorylate, put a phosphate group on another protein. And the protein kinase R, the R stands for RNase, so RNA. So if it detects the double-stranded RNA, it becomes activated. When it becomes activated, it puts a phosphate on certain targets. One of those targets is a protein called EIF2. So we've heard of that before, eukaryotic initiation factor 2. And EIF2 is a protein that's a GTPase. I've got a GDP drawn in the active site here, or in the binding site here, meaning that it has caused the GTP to go to a GDP at that point, and it has phosphate group. Another sign that you might be infected with a virus is that tons and tons of proteins might be trying to be translated and folded within the endoplasmic reticulum. And this can cause stress, because if all those proteins don't fold properly, then there are different quality control mechanisms that could kick in and say, hey, we've got too many unfolded proteins. We'll start the unfolded protein response, and we'll try to shut down host translation so that we can kind of get caught up on folding up all the proteins that are in the ER. And there you can lead to the activation of a protein called PERK. And again, we've got a K for kinase, and it has the same target. It targets EIF2 for phosphorylation. So we've got two different signs that that the cell has been infected with a virus, and it leads to the same final action, which is to phosphorylate EIF2. Well, there's a lot of EIFs to talk about, and so then we have to say, well, what was EIF2 again? And so here's a quick review. We've got our RNA in dark blue here on the cap, some 5' untranslated region, a start site, open ring frame, 3' untranslated region, and poly A tail. So in the previous video about translation regulation, we talked about these proteins that bind at the cap. We're going to skip over those for now because none of those are EIF2. Instead, last time we mentioned that there was a big complex called the 43S pre-initiation complex, or 43S PIC. And we didn't get into the details of what all the proteins were that were part of that complex. We did mention that the small subunit of the ribosome was part of that complex, but the other specific things weren't mentioned. We did mention that it would have a methionine tRNA, a tRNA that's charged with methionine. So the orange kind of upside down cross looking thing is a tRNA, and this is the initiator tRNA, and it's going to be looking for the start codon. So you remember AUG is the start codon, and that AUG codes for methionine. So we had said that this 43S pre-initiation complex would go bind, interact with these proteins that had begun a dogpile on the cap of the mRNA, and then from there, being at the very 5' end, it would start to scan 5' to 3' until it got to this AUG. So this pre-initiation complex has the ability to do some scanning. So the different EIF proteins here are EIF1, EIF1A, EIF2, EIF3, and EIF5. Okay, so there's a pretty good number of those. All the EIF4s were over here. So we got kind of a bunch of numbers besides 4 piling up on this 43S pre-initiation complex. So these are starting to scan for the start. Pile up there, so I've got an arrow going to recruit this whole complex to the 5' end of the gene, and then I've got an arrow here saying that we're going to scan until we find that start codon. In the previous slide, I made a big deal of talking about EIF2 being phosphorylated by those kinases PKR and PERK. So let's take a little bit closer look at that. So down at the bottom here, I've got the pre-initiation complex, and I've given it a little bit more space so that you can see that methionine charged tRNA here. And I also put a little bit of detail onto EIF2, and I show that it's bound. It's hard to read probably from on your screen, but it says GTP. So it's the GTP bound form of EIF2 is going to be the one that's here. So the EIF1, 1A, 5, and 3, those join later on. I'm going backwards on my flow chart here, but when you have EIF2 and it's holding on to a GTP, it can form a complex with the methionine charged tRNA, and that is going to be, those 3 things together are going to be the ternary complex. Maybe it's 4 if you count GTP and EIF2 as 2 separate things. So these together make a ternary complex. You bring in these other proteins. I appear to have forgotten to draw EIF3, but those all go together to form that 43S pre-initiation complex. Once that complex has found the start codon, a lot of the components dissociate and the large subunit of the ribosome gets on. And when they dissociate, we're not going to really track down what's happening to EIF1 or 3 or 5 or 1A, but instead we're going to take a look at what's happening to EIF2. So when it was first in the pre-initiation complex, it was the GTP bound version of that. Well, EIF2 is called a GTPase. It means that it can cause that last phosphate, so that's guanine triphosphate, cause that last phosphate to be cleaved off, and you end up with EIF2 bound to GDP. At this step, you would have an enzyme called a guanine exchange factor come in, and it'll put GTP in that site and pull GDP out. So it's putting GTP in, taking GDP out, and so that's the guanine exchange factor's job. So that then you have the EIF2 with the GTP bound to it again, and you can form a new ternary complex and recruit new things. And so there's just this circular recycling sort of thing happening here. But if this cell is thinking that it has a virus infection, we mentioned that these proteins, these kinases, protein kinase R and PERK, and there are some others as well, they can phosphorylate EIF2, and they do it at this point where you have the EIF2 with the GDP bound form, and you cause the protein to get basically stalled and fall out of that recycling recycling that we were seeing before, and it would stay in the inactive phosphorylated GDP bound form. Eventually, you might get a phosphatase come and pull that phosphate on, but if all the EIF2 in the cell gets phosphorylated and it's just stuck in this inactive form, what you end up with is translation inhibition across the entire cell. So that's sometimes called global translation inhibition. So virus infections, either through detecting the double-stranded RNA or through detecting the ER stress, leads to global translation inhibition. And it's with this inability for the cell to recycle all of its EIF proteins by putting that phosphate on EIF2. So there is global translation inhibition caused two different ways during a virus infection. All right, I think I'm starting to repeat myself, so we'll call that it. Have a good day. Like and subscribe or not.